Hello everyone. Today we are going to discuss about endothelins. So what are endothelins? So endothelins are basically a family of 21 amino acid vasoactive peptides. They are primarily produced by endothelium and are recognized as the most potent endogenous vasoconstrictor in human body. So these mainly act as vasoconstrictor. There are three isoforms of endothelin, that is endothelin 1, 2 and 3. These are the three isoforms in which it exists and each encoded by separate genes containing two disulfide bridges. Among these three, the ET1 is the most potent. It is the most potent endogenous vasoconstrictor. If we'll talk about the source and nature of endothelins, we'll come to know that, as we have discussed earlier, it is vasoactive peptide produced and stored in vascular endothelial cells and has important regulatory action in maintaining tone, vascular tone and homeostasis. We know this function. Three isoforms, as we have discussed earlier, 1, 2, 3, but one is found in endothelium, vascular smooth muscles, heart, lung, kidney and brain. Endothelin 2 is found in kidney and intestine, whereas endothelin 3 is found in brain, lung, intestine and adrenal gland. Moving further, now we'll discuss the receptor and its affinity. So, endothelin A and endothelin B are the two types of receptor in which endothelin 1, 2 and 3 will act. These are the three isoforms, but they will bind to two receptors, that is endothelin A and endothelin B. These are receptor types and these are the endothelin. So these are receptor A and B and these are endothelin which will bind to it, that is 1, 2 or 3. It will bind based on their affinity. So affinity of 1 and 2 is much higher than endothelin 3 for endothelin A receptor, whereas for endothelin B receptor all of them has similar affinity. Moving further on, we'll discuss the formation steps. So they are synthesized as pre-pro-endothelin 1 and then it is converted into pro-endothelin. This is pre-pro-endothelin mRNA, then it forms pre-pro-endothelin 1 which converts into pro-endothelin, which is big endothelin, which is an intermediate and which forms endothelin. This is the endothelin. The endothelin is converted to ET1 by endothelin convertase enzyme. So these are the enzyme, endothelin convertase enzyme ECE1 and 2, which convert the intermediate into endothelin 1 and these act on the receptor and perform its action. Same goes for 2 and 3. Here you can see the ETA receptor will have GQ and GS action and ETB has GI and GQ action. As we know, when GQ gets activated, it causes increase in IP3 and DAG pathway, which will increase calcium, which leads to vasoconstriction. This you have to keep in mind. Whereas if we'll talk about GS will increase CAMP and GI will decrease CAMP. These are the three functions of GQ, GI and GS. Now we'll discuss where are these receptors present. We know there are two types of receptors, but where are they present? ETA are mainly present in vascular smooth muscle cells. These are the vascular smooth muscle cells, these blue one. These are tunica media, the middle layer of endothelium. And ETB is present on endothelial cells, which is the innermost layer, which is tunica intima, and in vascular smooth muscle, that is tunica media. So ETB is present on both and ETA is present only on tunica media, that is vascular smooth muscle cells. ETA function is to cause vasoconstriction, whereas ETB function is to cause vasodilation via releasing NO and PGI2. This is a more detailed diagram. Here you can see where there are various types of stimuli present over here, which increases, causing positive impact on ET1 gene, and these are the inhibitory stimuli which inhibits this. If this got stimulated, that is pre-pro-endothelin and endothelin, it causes formation of endothelin, big endothelin, which is intermediate and which forms endothelin 1. These endothelin 1, as we have discussed earlier, will act on two receptors, ETA and ETB. As we have seen, ETB is present on both, that is vascular smooth muscle cells and endothelial cells. So ETB is present over both these surfaces, whereas ETA, A and this is B are present on both the surface, whereas ETA is present only on vascular smooth muscle cells. So when ETB is present on vascular smooth muscle cell, it will cause vasoconstriction. But when ETB is present on endothelial cell, it will cause nitric oxide release and cause vasodilation. So ETB has two roles based on its location. If ETB has two roles, if it is located on vascular smooth muscle cells, it causes vasoconstriction. If it is present in endothelial cell, it will cause vasodilation. So where we have this endothelin, where we will see this endothelin in action, mainly we'll see them in pulmonary arterial hypertension. So pulmonary arterial hypertension means when the mean arterial pressure of pulmonary arteries are more than 20 mm Hg at rest and measured by right heart catheterization. There are different types of pulmonary hypertension, but our main focus is on group 1, which is caused by arterial hypertension. In this, the endothelin acts, so we'll focus only on group 1. If we'll talk about the pathophysiology of pulmonary arterial hypertension, there are 3 pathways. It increases endothelin pathway, that is, it causes vasoconstriction and proliferation. It decreases nitric oxide and it decreases prostacyclin. So here we have different types of drugs which will be used in pulmonary arterial hypertension. These are the important drugs. We have Bosentan, Macitentan and Ambrisentan. Ambrisentan is selective for ETA receptor antagonist, whereas Bosentan and Macitentan will have dual ETA and ETB antagonist. These drugs are used in pulmonary hypertension. These 3 drugs, Bosentan, Macitentan and Ambrisentan. You can remember it as Ambrisentan as A, so it is selective for ETA, whereas B and M has dual action on ETA and ETB. Now we'll compare the roles, the features of the drug. If we'll talk about Bosentan, it has dual action. Macitentan, same dual action. Ambrisentan, ETA antagonist selective. Indication, pulmonary arterial hypertension, same goes for other 2. Dose 6.25 mg twice daily and increases up to 125 mg twice daily for Bosentan. For Macitentan, it is 10 mg once daily. For Ambrisentan, it is 5 mg once daily and maximum up to 10 milligram. Half-life is 5 hours for Bosentan. For Macitentan, it's 16 hours for parent drug, and for active metabolite, it's 48 hours. And for Ambrisentan, it's 9 hours. Moving further, if we'll talk about the metabolism, Bosentan is metabolized by CYP3A4 and CYP2C9, whereas Macitentan, it is metabolized by hepatic metabolism with active metabolites. So it is metabolized by liver. Ambrisentan is metabolized by CYP3A4 same and CYP2C19 and plus glucuronidation. Major adverse effects are hepatotoxicity, edema, anemia and headache. For Macitentan, it's edema, anemia and generally better tolerated. For Ambrisentan, it's peripheral edema and headache. If we'll talk about hepatotoxicity, common for Bosentan, it requires regular LFT monitoring, and for Macitentan, it is less frequent. The hepatotoxicity is less frequent. And for Ambrisentan, it is much less common than Bosentan. Drug interaction, there are many drug interaction for Bosentan as it has CYP3A4 and CYP2C9 inducer. For Macitentan, it has fewer interaction, and for Ambrisentan, it has relatively fewer interactions. If we'll talk about special precautions, so first drug, that is Bosentan, is teratogenic. This drug is teratogenic. It is contraindicated in liver disease. Second, Macitentan, caution in hepatic impairment. We have to keep that in mind. Ambrisentan is contraindicated in idiopathic pulmonary fibrosis. Moving further, these are some newer drugs. That is Ambrisentan. This drug has dual endothelin 1 receptor antagonist, ETA and on ETB. It is mainly used for resistant hypertension. So you have to keep that in mind, that Ambrisentan is used in it is approved by US FDA 2024 for treatment of resistant hypertension. Next drug we have Sparsentan, which has endothelin A plus angiotensin receptor antagonist, and this is used for reducing proteinuria in IgA nephropathy and focal segmental glomerulosclerosis. This is the new finding demonstrating anti-proteinuric effect and potential kidney protective action. Next we have combination therapy in pulmonary arterial hypertension, in which there are systematic reviews that indicate that improved outcome when endothelin receptor antagonists are combined with phosphodiesterase 5 inhibitors like sildenafil. It shows better hemodynamic and functional status than monotherapy. So these are some drugs that we have discussed. I hope you guys have find this video helpful. Today we have discussed about endothelins. Endothelins, we have discussed, it has 1, 2, 3 isoforms and they act on different receptor like ETA and ETB. ETA are present in vascular smooth muscle cells and ETB are present in endothelin and vascular smooth muscle cells. ETA has main role in vasoconstriction, whereas ETB has main role in vasodilation. If we'll talk about the role, the action of ETB, it has different role. If it is present in vascular smooth muscle cells, it causes vasoconstrictor action, constriction, and if it is present on endothelin, it will cause vasodilation. Then we have studied there are antagonists of endothelin, in which we have studied Bosentan, Macitentan and Ambrisentan. These are antagonist drugs which are used mainly in pulmonary hypertension, because in pulmonary hypertension there is increase in endothelin. Because there is increase in endothelin, so we will need the antagonists like Bosentan, Macitentan and Ambrisentan. And there are some recent advances which we have discussed. I hope you like this video. Kindly share and subscribe our channel for more such videos.